Hey, how's it going everyone? It's Chow here. So I don't have a lot of time, but I thought I'd put out this video anyway so we can talk a little bit about translation. So let's get started. So translation is this very interesting process where we're trying to get an mRNA strand into a protein, or more specifically, we're trying to read an mRNA strand to get a protein out of it. So there needs to be some components of translation that will allow you to get you there from just a simple mRNA strand to a complex protein. So the first component that we're going to talk about is the codon. Now the codon is a very interesting section of the mRNA because it's talking about bases on the mRNA, or more specifically, three separate bases on an mRNA, and these three separate bases together code for an amino acid. It doesn't have to be a specific amino acid per se, but there is going to be one codon, which is three bases, that code for an amino acid. But sometimes there's a little bit of give or take in that there are different codons that code for the same amino acid, and we'll talk a little bit more about that in the next slide. But for now, let's just look at one specific codon, and that's AUG, which codes for methionine. It's very important because this is the start codon, so when translation happens, methionine is, generally speaking, the first amino acid. So when a ribosome comes in to recognize a certain start zone within an mRNA strand, it always looks for AUG, which again codes for methionine. But you have your strand of mRNA. However, the issue now is you don't have the amino acid. But what carries that amino acid, of course, is pretty important. But before we move on to that, we have to talk about other things, and that is designations. So again, three bases will designate an amino acid. That's a codon. Now, if you want to move on and do a little bit of mathematics, you'll figure out that four bases in mRNA, so A, C, G, and U, remember uracil replaces thiamine in RNA compared to DNA, and there are all these per three base codons, which means that 4 to the third is 64. So there are 64 possible codons, but there are only 20 amino acids. So again, as I mentioned just a few seconds ago, there are redundancies within the systems, and you can see something like this. For instance, alanine over here, GCU codes for alanine, GCC codes for alanine, and so on and so forth. So that's really cool because it allows for a little bit of redundancy, and as well as this other thing called the wobble effect. So wobbling is that specificity for the last base of the 3' end of the codon is not always observed. So you have instances like this in alanine where the last base is not really important. So as long as the ribosome recognizes the first two bases, it has a pretty good idea that what it needs to put in is it needs to recognize and put in an alanine there. So in a way, you can think about it as it kind of allows for a little bit of room for error, maybe. That's one way you can think about it. So it decreases the specificity in some instances. So that's the wobble effect. One thing you should know, however, is how to read this table, and I will probably put up another video teaching how to read this table, but there are no guarantees because, again, I don't have a lot of time. So let's move on. So if you look at something like this, this is the wobble for leucine, and you can see that you have your mRNA over here, and then you have the anticodon, which is something that I think we should talk a little bit about, on the tRNA, and the tRNA is over here, the transfer RNA, and it has anticodons that correspond to the codons on the mRNA. So CUC here is the codon, and GAG is the anticodon. Notice that this is still anti-parallel. And you have instances like this where you can have sort of a wobble pairing where the anticodon in this instance is GAG, but you're still able to pair because, frankly, the first two letters match. And so it allows for, again, a little bit of room for error, and allow again for that instance where even if there is a slight change or an issue within the mRNA strand, you still have an increased chance of putting in the correct amino acid. So this is wobble for leucine. So I mentioned a little bit about other things aside from codons, but before we move on to the transfer RNA itself, I think we should talk a little bit about the stop codons. Basically, stop codons don't code for any amino acids. It just kind of tells the complex to stop translation. There are 3 different stop codons. You should probably memorize these, so UAA, UAG, UGA in the 5' to 3' direction. And these again tell the complex to stop translation, and they don't code for amino acids. Cool? So let's move on to tRNA or transfer RNA. So transfer RNA is a pretty cool component. It's another component of translation. And what's really interesting about tRNA is that it's what carries the amino acid. So if you get asked a question, what carries the amino acid before translation actually occurs, it's the tRNA. And more specifically, it has a 5' end and a 3' end, and the amino acid attachment site is always going to be on the 3' end. Now, tRNA is really cool because it carries the amino acid, but it also has a 3-base sequence called an anticodon, which corresponds to the opposite bases on a codon on the mRNA. So if the codon is AUG, the anticodon is UAC. So that kind of tells the strands and the whole complex what needs to be added. So the correct sequences coded by the codon will be matched with the anticodon, which is part of a tRNA that has the correct amino acid. Hopefully that kind of makes sense. But just a basic overview, I mean, when you look at a transfer RNA or a tRNA, it has a primary structure, and the primary structure is just kind of a linear sequence. The secondary structure is sort of a clover leaf shape like this. And then if you look at the tertiary structure, it's more folded, and there are different interactions, so it no longer looks like a clover, but more like sort of an L-shaped. So that's the basics on transfer RNA. But before you actually can have a useful transfer RNA, you need to charge it. And what I mean by charging is basically using an enzyme. In this case, it's called a tRNA synthetase. This is a purple thing over here that will basically allow for the attachment of an amino acid onto a specific tRNA using a specific tRNA synthase. So if we have a situation like this, you have a specific amino acid, and then you have your tRNA synthetase. You use the hydrolysis of ATP as well as the combination of that amino acid into one of the sites on the tRNA synthetase, and 2 of the phosphates leave, and you're left with AMP as well as the amino acid within the tRNA synthetase. Then you have an alanine or whatever amino acid specific tRNA come in. Remember, there are specific tRNAs that come in and sort of bind to this complex. The AMP leaves, so you have this kind of tRNA bond formation. So you have this bond formation of that tRNA molecule, this green molecule that comes in to the amino acid that's within the tRNA synthetase, and you ultimately have a charged tRNA. So again, there are specific tRNA synthetases and specific tRNAs. And for example, you can use the 3' end, the OH of the 3' end, and maybe you use like an alanyl tRNA synthetase, and then you attach an alanine to that tRNA, and now that tRNA is charged with alanine. So that's how you charge a tRNA. Remember, charging isn't about pluses or minuses per se, but it's just simply about charging a tRNA molecule, so now it has the amino acid, so now it can be useful in a translation process. So that's pretty interesting. So next we have the ribosome, which is a very important part. It's kind of the last part, I guess, of this whole translation complex. But the ribosome is important because it's the molecular machine that actually does the translation, and it also is the machine that sort of finds those correct amino acids and binds them together. So it's very important because of what's known as peptide bond formation, or bond formation between two amino acids. And when you look at a eukaryotic ribosome, you have a large subunit and a small subunit with differences in the amount of ribosomal RNA as well as the number of peptides. But it's really important to know that, generally speaking, the ribosomal subunits don't actually come together except when synthesis is actually needed and or during translation. So during translation, the subunits join together, and then when translation is over, they separate. So that's really cool. So now we can go through the steps of translation itself. First step is initiation, so starting translation. And that usually happens when a small subunit will bind to an mRNA, and that subunit will start kind of reading it and figuring out where everything is. And then eventually the subunit will stop at a certain region, more specifically the region that is with the start codon, and then you have a certain tRNA that comes in. And if you can guess which one it is, I think you have a pretty good idea, because it would be the met tRNA or methionine tRNA. And the tRNA with the methionine connected to it will recognize the AUG site with its anticodon, the UAC anticodon, to the AUG codon. Keep those terms separate. And it attacks via the P site again. So there are 3 different sites on the large subunit of the ribosome. So it attaches to one specific section, but let's keep things in order and say that when it comes to the attack, the tRNA attaches to the mRNA strand as well as the small ribosomal subunit, and when the large subunit comes in, it puts the tRNA with the methionine attached at the P site. And this, of course, will lead to translation to initiate. So let's just keep in mind that there are 3 sites again, and get them separate. We have the A site, which is where things sort of come in later on. We have the P site, where the peptide bond formation actually occurs, hence the letter P. And then E, which is the exit site, where the tRNA leaves. So you can think of it using like using APE, for instance, APE, APE, and you'll know that the direction is from the A to the P to the E. But when it comes to initiation, the large ribosome actually binds directly to the small ribosome as well as the mRNA, and it puts the tRNA that's already bound to the complex in the P site. So that's, I guess, a little bit of a difference. If we move on to the next part, and that's elongation, we actually have more things happening. So the next charged tRNA is going to come in, recognize the next codon present in, for instance, the A site, and in this instance, the codon present, let's say, is CCG, and that codes for proline. And so the proline is going to zap in along with the incoming tRNA that has the proline attached to it, and it's going to recognize the site, and it's going to go in and bind. And then there's going to be peptide bond formation once the tRNA is within this whole complex. So the large subunit will catalyze peptide bond formation, and the methionine will actually join to the proline. And the mRNA then, of course, moves, and the tRNA originally linked to the methionine will get moved to the E site, and then it's going to get ejected. The tRNA, which has the proline attached, gets moved to the P site, so everything kind of shifts over. And because the E site is the exit site, once that tRNA that originally had methionine bound to it is in the E site, it gets ejected out. And so this process just repeats and repeats and repeats. And I think you move on and on and on. So tyrosine comes in, recognizes the sequence, and it just is a repetition of what happened before, where you just have something, the codons moving into the A site, and then something else, the tRNA, recognizing that, oh, okay, this particular codon sequence matches the anticodon sequence on myself, and then it has that particular amino acid bound to it, and so on and so forth. And so you catalyze bond formation until you get a relatively long strand of amino acids. So remember that amino acids are formed from the N-terminus to the C-terminus, and you have the start over here, the N-terminus with methionine, and then you have proline, tyrosine, and this is just an example. That's not always how the starting that's not how all the starting amino acids always are. It just depends on what the sequences are. So you have this instance where you have methionine and this growing strand. Eventually, termination occurs, and termination is basically the stoppage of translation. And translation stops when it reaches a stop codon. So here we have UAA as an example. Remember, UAG and UGA are also stop codons. But you have UAA over here, which is a stop codon, and it reaches the A site, and it kind of causes a release factor to bind into this A site. When that happens, it causes everything else to be cleaved and ejected. So the peptide is released, cleaved from the last tRNA, and the tRNA is also released from the ribosome. So now you have this amino acid polypeptide. And once that occurs, everything is kind of finished, so the mRNA is also released from the ribosome. The release factor is also released, and the large units and subunits kind of separate themselves and now are no longer attached to the mRNA because translation has finished. So again, important note to keep in mind is that protein is made by reading mRNA in the 5' to 3' direction. So this is a little bit different because in, for instance, let's say transcription or DNA synthesis, the reading is from the 3' to 5' direction. But in this instance, you're reading, remember, from this end to that end, so you're going right, and the 5' to 3' end is also corresponding to that. So if you look back to something maybe like this in initiation, you have the 5' here and the 3' here, and the reading is actually going in that direction. So it's a little bit different than transcription as well as DNA synthesis. One other thing to keep in mind is that translation often occurs in a long series in a way where you have multiple ribosomes attached to the mRNA and basically cause translation to occur all at once. And that creates what's known as a polysome, where you have just a bunch of ribosomes attached to this poor little mRNA molecule, creating more and more and more groups of proteins. And this polypeptide sequence, which is now sort of a new protein, can then be changed and edited in many ways. But by having multiple ribosomes attached to the mRNA, you can increase the translation as well as the translation product. So you can increase the amount of protein output simply by having more ribosomes read and synthesize and attach themselves to the mRNA. So I hope that kind of made sense. And then, of course, after translation, polypeptides are often edited. There's glycosylations, there's sometimes proteolysis, and a lot of different things that can happen to edit the protein before it becomes a final product. But that's basically it for translation. I hope you found this useful. And I guess happy studying, everyone, and take care.